I have the honor of introducing Dr. Ferrucci today. And Dr. Ferrucci is a geriatrician and physician scientist. His area of focus is in the disablement process, physical function, and mobility. He has played a key role in the development of major longitudinal aging studies, including INCANTI and also the Baltimore Longitudinal Study of Aging. He is now presently the scientific director for the NIA, and he has over 500 peer-reviewed articles in peer-reviewed journals. I had the honor of knowing him as a geriatrics fellow in Baltimore, and we're quite honored for him to come and speak to us and give him his perspectives on the BLSA. Thank you, Dr. Ferrucci.
Well, thank you very much for the nice introduction, and I'm really pleased to be here. And my visit so far has been incredibly pleasant, and I met really fantastic people that are interested and enthusiastic about study aging, which is really my passion. And what I'm going to tell you today is that I am a really lucky guy, because the thing that I'm doing is because I've been given incredible opportunities to work in studies that in most cases were created before I was there. So I just took this very beautiful tree that was producing these incredible fruits and take the fruit and eat them. And that was very nice and very pleasant and resulted in many papers. But most of what you see in my CV is really work that has been done in collaboration with others. So I'm going to talk to you about the Baltimore Longitudinal Study of Aging. Before I do that, I need to tell you how I got here, because that's very important to explain what is the paradigm that I'm trying to implement in the BLSA. And to make a very long story short, this picture really tells you why I'm so interested in mobility. And I'm so interested in mobility because this is the typical trajectory of mobility that human beings experience over the lifespan. And so there is a very, very rapid increment in mobility, some sort of a stability over adult and age, and then some sort of accelerated decline that result into disability. And of course, there is incredible heterogeneity in this trajectory, and this only represents the average in the population, and there are some lucky people that get to very, very old age, and they are still very highly functional. And I will remember always that when I was much, much younger, I was doing a study of centenarians in Sardinia. I was actually the interviewer. And so I was going to the different cities, interviewing these old centenarians. And one of them told me that they couldn't really spend time with me because he had 3 children. All 3 children had families. They were working in the morning, so they didn't have time to shop. So he was shopping every morning for 3 families and carrying those bags from the shops and getting on the stairs, getting the bags in the home. And the only way I could interview him was really going there and helping him with the bags. And during helping the bags, he will respond to me and addressing the interview. So there are exceptional people, but I think that heterogeneity is not a bad thing. I think heterogeneity is our windows of opportunity. If there was not heterogeneity, if every single person was doing something like this, there's really little that we could do. I mean, it would be such a fixed system, then changing that system will be, I wouldn't say hopeless, but we will have very little hope. We know that because there is heterogeneity, because there are exceptional people that can do much better than that, then maybe we can push the curve and we can push many more people to be highly effective, both physically and mentally, over the lifespan. So this is probably the most important landmark paper that I ever published. It's probably not the most sophisticated, But is the paper that influenced what I have done in the last 2 decades. So in 1995, I was working with Jack Guralnick, which has been my mentor, and we implemented a very simple set of performance measures in the established population for epidemiological studies of the elderly. We were doing that. We were not understanding the value and the effect that implementing those simple tests will have had on our career and also in the evolution of the geriatric field. And so what we were doing is we wanted to have a quantitative measure of mobility. Everybody was studying mobility from the point of view of interviews, and so there is a lot of variability. We said, let's implement something simple. And when you work in large populations, something should be easy, simple. You don't need training. You don't need equipment. It needs to be cheap, because otherwise it cannot be implemented in a epidemiological study. And we created the SPPB, which many of you know what it is, but I'm going to tell you in very simple words. So one thing is that you make people walk for 3 or 4 meters. At that time, it was 2.8 meters. The current invention is 4 meters. And then you measure how much time it takes to walk for 4 meters. And then you make them rising from a chair 5 times without using their arms. And then you measure how much time it takes to do the entire task and whether they're able to do it or not. And then you ask people to maintain their balance and in progressively more challenging positions. And when I say more challenging is when you have to maintain balance, you're very good at maintaining balance in the anterior posterior position because your feet are in this direction. But if you restrict the base of support, you're not very good in the lateral direction. And so you restrict the base of support progressively, and you see whether people are able to maintain balance for 10 seconds. So you do side by side, semi-tandem, and then tandem, one foot after the other, and then you see whether people are able to do that and maintain that position for 10 seconds. So we apply that in the entire population of the EPI studies. We divide their performance into quartiles. And to make a long story short, we created a score from 0 to 12, which was a perfect score and inability to do all 3 tests. Then we measured that in the East Boston subcomponent of the EPIs at baseline, at follow-up 6. And then we got a score. And then we look at these same people 4 years later. So if you select at baseline those participants that had no mobility disability and no ADL disability, and then you measure the incident, so the new developed mobility disability and ADL disability, you construct something that is like that. So you have a score from 4 to 12. You don't have below 4 because you cannot be non-disabled and have a score lower than 4. So at baseline, you are a population at risk. And then you measure mobility disability. And you can see that a score of 4 was predicted at 4 years of developing mobility disability in 50% of the population. In the score of 12, only almost less than 3% of the population developed mobility disability. Now, that is incredibly powerful. I mean, there is no one single test in medicine that is as powerful as that. If you think of measuring blood pressure, cholesterol, it's powerful. It's really not even close to the predictive value of the simple test that cost, $3, need no expertise, no technology, is immediately interpretable. So this is a paradigm that we use to study mobility. And this really comes from when in another life I was a clinician. And so that we had a clinic on mobility, and that is the way we were thinking about looking at our subjects. So we thought that mobility starts in the central nervous system and is motivation. So it comes from the frontal cortex. If you don't want to move, you end up not moving. And then you create a motor program and then you create this motor program. You wire them through the basal ganglia and then through the spinal cord, and then you use the peripheral nervous system to activate the peripheral effectors for this motor program, which are the engine of the body, which are the muscles. And then you have moving what is the hardware of the body, which the bone and the joints, and that you need energy to do that, because you don't energy, you can't do that, and then you need feedback from the environment. Because you're now living in a vacuum, so if you don't pay attention to how the terrain is changing with you, you're going to fall down very, very quickly. So vision, proprioception, hearing and tactile sensitivity are very, very important. So this is a study this is a paradigm that was actually implemented in the Chianti study. When we started doing analysis in the Chianti study, we realized something very interesting. In the longitudinal analysis, those quantities were changing harmonically. So the people that tended to have the most change in muscle strength were also the people that were likely to have decline in cognition. They were also the same people that started to have energy problem. So there some sort of underlying latent variable that was explaining decline with aging across multiple systems. So when I started the BLSA in 2002 and I needed to redesign the study, I used this part of the Chianti study, but I also added a layer of measures that were going to mechanisms, so that I wanted to see whether there was a problem of oxidative stress, there was a problem in the autonomic nervous system, there was some pro-inflammatory state, whether the hormones were altered, and whether there was a problem in nutrition or a problem in physical activity. And the paradigm at that point was the following. You have energy going in, which is nutrition and oxygen. Your energy going out, which is physical activity, mental activity, and resting metabolic rate. And then there are a number of systems that really shift the energy from one place to another so that you maintain the homeostatic equilibrium. But then, after a few years, we felt that we could not really explain frailty. We could not really explain the interface between frailty and disease. And one of the problems that still come from my experience as a clinician has been always that frailty is such a neat and nice concept as a scientist that makes a lot of sense clinically. But every time I was trying to put together, frailty and disease in my patient, I couldn't really do that. So the problem I had is that, yes, I can screen for frailty using one of the many systems and many criteria I have, but then when I've done that, what do I have? What I'm going to do about that? How knowing about frailty is going to help me curing for that patient? And that is, to me, why the beautiful concept of frailty have never really been translated into geriatric medicine. So I needed to address that. So we had a lot of discussion, and I'm going to make a very long story short. And the story starts with this. Let's assume you are in Perugia. Perugia is a beautiful city. That there is a square in Perugia, and you're walking around, you're looking at people going around, and then you see that there is a woman on the side, and you start thinking, well, how old is that woman? And your supercomputer in your brain start clicking, and you have a lot of information that are going in, and the way she walks, the expression, the type of the skin, and the hairs, and many, many, many things come into your mind. And then at some point, click, you have a number like 42, 85, or 57, or 62, or whatever is the number that comes out. But you don't really know how that number is coming into your brain. That is a fundamental problem. It is a fundamental problem because we are studying aging, And we don't even know how to measure aging. So how do you know that something has accelerated aging? And if you slow down aging, how do you know that you have slowed down aging? And so mobility was a very nice paradigm, but when you lose mobility, it's a little bit late in the pathway. It's really when all your compensatory system have been activated at 100%. And that is, when you really lose mobility, there is not very much you can do. It's very, very difficult to intervene. So I came up with some domains that, to me, describe the aging phenotype. And that is the model that I'm going to talk to you about today, and I'm going to show you data about. So you can describe the aging phenotype in humans in basically 4 domains. And please, if you have any other suggestion, and you think that this domain do not exhaustively explain the aging phenotype, I'm very, very happy to discuss and to implement them because I'm looking for suggestions. So the first domain is changing in body composition. Whatever we do, even if our weight is stable, you know that our body changes, and I'm going to show you data about it. Then the second, and to me the most exciting part, is that the energetic change. So both the production of energy and the utilization of energy become very, very, very different with aging in almost every individual. Then you have a problem of homeostatic dysregulation, and I'm going to talk a little bit about it. And then you have neurodegeneration. If you pull together all those 3 aging phenotypes, you really are summarizing the most important phenotypes you see with aging. So that seems to be, simple, but it's incredibly helpful to me. Because on one side, you can think about frailty as the overall and massive changes and deterioration across multiple aging phenotypes. And then you can also start thinking about disease as determining the physical and cognitive decline with aging through their effect on the aging phenotype. Somebody was talking to me about diabetes and frailty. If you think about diabetes, there is clear demonstration that diabetes determines accelerated decline of lean body mass and muscle strength. There is a strong effect on energetic, but also more recently on efficiency of energetic use during walking. There's clearly primary on homeostatic dysregulation, and there is neurodegeneration at the peripheral and central nervous system. And now we accept at epidemiological level that diabetes is the strongest factor for dementia. So disease can be conceptualized as interfacing with the element of frailty through the aging phenotype. And so in a way, aging becomes almost a comorbidity. And you can understand how the 2 fit together. There's another, to me, very, very interesting clinical aspect that could help us. You have a number of conditions in geriatrics that are called the geriatric syndrome. And they are called geriatric syndrome for the absence of a better name, because they are multifactorial, because they can come in many, many different ways. But if you think about it, the aging phenotypes, a combination of them, are really responsible for all the geriatric syndrome. If you think about urinary incontinence is a combination of reduced muscle mass and muscle strength and neurodegeneration. If you think about disability, it really comes from these aging phenotypes. If you look at delirium, for example, certainly is an issue of the neurodegeneration and reduce ability to cope with the psychological and environmental stress. So in a way, understanding the aging phenotype will give you the key to interface the clinical situation of your patient with the disease but also understand that if you want to reduce the effect of disease on disability and frailty, you need to act on the aging phenotypes. So give you also a target for intervention. And in the next part of my presentation, I'm going to show you data that were collected in the BLSA for each one of these domains. I'm going to start with changes in body composition. So when you're lucky and you work in the BLSA, and there are a lot of people that have worked for you in collecting data, you get 50 years of data collected on body composition. Your trajectory of the span over 40 years, people that have been evaluated 15, 16 times, unique data, so you can start drawing this trajectory. And you know that those trajectories are right because they're not biased by your cross-sectional data. So you see that in the BLSA, at least, weight increase with aging, both in men and in women, and start declining around between the age of 65 and 70. And this decline tend to be accelerated with aging. That decline in weight does not occur in all the individuals, but in many of them. And we are now thinking that, in fact, the decline in weight is due to disease. It's not primarily related to aging, because there are some individuals that actually maintain stable their weight up to very, very late in life. This change in weight is really hiding substantial change in lean body mass and in fat mass. With the lean body mass that increased up to the age of 35 to 40, and then a very small constant decline, and then accelerated decline after the age of 65 to 70. Fat mass increase and then decline in parallel of the decline in weight. And similarly, you can see what's happening in women. It's almost the same thing. Now, what is interesting here is that on one account, there seems to be two very, very important periods in life. One period, it's really here. So maybe one way of addressing the trajectories is start to do intervention in people that are in their 40s, if you want to have an effect later on. And then there is something else that is happening around the age of 70. But if you look at the fat, you also see that not all the fat is the same. So if you look at subcutaneous fat, and these are data from the CT of the abdomen that were collected in BLSA participants, clearly the trajectory is the trajectory I showed you before. But the visceral fat, which is the fat that is metabolically active and is the fat that is making the travel, it's really never going down and seems to be increasing with age in almost every participant that we have seen. And you will see that that increase in visceral fat is actually very, very important from some of the aging phenotype. So let's go back to muscles. So I told you that there is a decline in lean body mass with aging, but the decline is in part due to aging and in part due to disease. So here in the BLSA, you look at percent with low grip strength as a measure of sarcopenia. And you see that those in the BLSA with COPD tended to be having the same prevalence in younger age. But when you go to older age, there is a cumulative effect, a synergic effect of aging and disease that has an effect on the aging phenotype. If you look at diabetes, the effect of diabetes is even more evident. You see that those are knee extension torque, so a direct measure of strength. You see that there's actually increment up to the age of 30, and then progressive decline, but there is a divergence in the trajectory of decline that is much more accelerated in older people. So the effect of diabetes becomes synergic on the effect of aging. Okay, so of course the change in in body composition also affect metabolism. And here, we looked at the quartile of mid-thigh muscle cross-sectional area from CT, and then we look at our data on the glucose tolerance test. And you can see that in those in the highest quartile of cross-sectional area, you have much better glucose metabolism, and insulin sensitivity measured with the Matsuda index was much better. And that is to be expected. And even when you look at obesity, and you classify people in lean, only central adiposity, central global adiposity, and global adiposity, you can see that in those with central and global adiposity, you have the area under the curve of the glucose tolerance test seems to be much higher. And when you divide that into the early stage and the late stage, the early stage being due to a dysfunction in the liver delivery of glucose, and the second instead being a more direct measure of insulin sensitivity at the level of the muscle, this seems to be affected much more in those with central and global adiposity. Nothing really surprising here, although has never been done really before in the larger population. But the effects are not only on metabolism. So one of the things we wanted to know is whether the change in body composition that occur with aging also have an effect on cardiac function. So one of the things that occur with aging is that you have a problem in diastolic dysfunction. This, for example, is an echocardiogram with a Doppler flow through the mitral valve in a typical subject. And you can see that there is an increased flow here because of the entry of the blood in the atrium, which is a passive phenomenon. And then you have the atrial contraction that increased the flow dramatically to fill the ventricle even more. And the ratio between these two, as you can imagine, is a measure of how much relaxation you have in the ventricle. And so you can measure by an EA ratio what is the relaxation ability, which is the diastolic dysfunction of the ventricle. And when you look at this in the Baltimore longitudinal study of aging, you see that the EA ratio tend to decline with aging. But we don't know whether this is a direct effect of aging or there is something else that's going on. So we did recently an analysis where we looked at the effect of body composition. So this is the beta coefficient that is expressing the relationship between aging and this diastolic function parameter. And so when you adjust for total body fat, you see that the effect of aging is not affected at all. But when you adjust for visceral fat, you see that there is a substantial reduction in the coefficient, suggesting that there is some indirect effect of visceral fat that has an effect on cardiac function, so that part of the diastolic dysfunction that you see with aging is due to the change in body composition. So not only the body composition change have an effect on metabolism, but also affect cardiac function.
Are these relationships based on simultaneous data?
Yes, yeah, absolutely. Exactly. So they have been measured, they are collected at the same time in the same subjects. So that's allow us to do this analysis because of that. So one thing that I want to tell you is that so far I'm only talking to you about the body composition in terms of muscle mass. So this is the muscle cross-sectional area, decade by decade in the BLSA, and this is the blue one. But when you look at the change in muscle strength, instead of looking at muscle mass, there's a lot more change in muscle strength than there is change in muscle mass. So that aging is also associated with the change in muscle quality. You have less muscle, and the muscle you have is less functional. So that, it seems to be trivial to say, but let's say you have the same cross-sectional area, and then you look at the same people with the same cross-sectional area. Somebody has this strength, somebody has this torque, somebody has this torque, and this can be as important as being disabled and non-being disabled. So we need really to understand what's going on if we want to be effective in changing the muscle strength and preventing disability. This is incredibly important because remember that all the intervention that have tried to improve muscle mass, almost all the intervention, the testosterone, the SARMs, has been totally ineffective in changing disability. You have an effect on muscle mass, you have an effect on muscle strength, very, very small. That never translates into any clinical effect on disability. So we need to find ways not to act on the mass, but to act on the muscle quality. And this, you've probably seen this slide several times, but what I want to show you is that yes, if you take women that have the same BMI, there's a muscle that is very well represented with very little subcutaneous fat in the femur here. The fat changes and increase even more in the 80 years old. But what is really important here is to see that there is also some sort of destructuration of the muscle fiber. And even the muscle that remain, that don't seem to be affected, have a much lower density. So there is something that is changing intrinsically in the myofibers. And what we suspect, and we have some preliminary data showing that, is that what is really changing is the amount of fat that is into the cells. So let's say that you're a clinician, and then you see that there is not a problem of muscle quantity. What could be the problem of muscle quality? And there are basically three different ways of looking at that. One is to look at muscle composition, cell type, protein contents, lipid content, muscle energetic, and neuromuscular controls. And in the BLSA, we are now measuring each one of them. So, for example, these are the proton spectroscopy. When you do proton spectroscopy, you have to target a muscle that is attached to a bone so that it doesn't move very much during your analysis. And you obtain a spectrum that is something like that, with a split in the middle. And because of the orientation of the lipids between the cell, then within the cell, you can really divide what are lipids that are in the extracellular environment and lipids that are the intracellular environment. And for example, here you see that people with higher BMI also tend to have much more intramyocellular lipids, suggesting that in some way is obesity, and I don't have the slide here, but also aging that is associated with change in intramyocellular function. And these are just preliminary data, but I can tell you that there is we have done very few muscle biopsies, but when you do muscle biopsy, you find that the amount of intramyocellular lipid measured with proton spectroscopy is strongly correlated with the mitochondrial efficiency in ATP generation. So let me get to the energy imbalance, which for me is the most exciting part of this presentation. So these are data from Ed Licata group that were published years ago. I have nothing to do with this data. Was published when I was not, they were produced when I was not in the BLSA, and so I was not part of this paper. But show clearly that if you measure longitudinally exercise capacity with a treadmill, there is a decline. And when you look at the decline as % decline compared to the previous decade, there is accelerated decline of fitness. And VO2 max decline with aging. The problem I always had with this slide is that nobody works the entire day in VO2 max. We all work and we do little things that sometimes for very few minutes are VO2 max, but we are really a submaximal people. So Studying VO2 max, which is something that has been done in the past, is really not enough. We need to do a lot better. So then take us to understand what's going on. What is going on in the different types of energy that we use in the day? So the way I want to talk to you about this is that imagine that this is the entire energy that we use in the day. And that this is a box of energy that you get in the morning. There's a box of energy that you're given, and you have to live with that box of energy. You need carefully to consume that box, because if you use the box before the end of the next morning, when the mailman comes with the next box, you're going to die. You need to have that energy and save that for the next morning. So a large part of this energy is actually resting metabolic rate. It's just the energy for life. You need 60 to 70% of energy just to stay alive. And when I read that, I was shocked. I did not believe that. But actually, if you think about it, it makes a lot of sense. Because we are creating a couple of billion of red blood cells every day. You're doing protein synthesis. There are 2 million of apoptosis in the muscle. I think that when you consider that, you need a lot of energy to stay alive. And believe it or not, 1/3 of this energy is used to maintain a sodium-potassium gradient in the cell membrane. Is a sodium-potassium pump that consumes 1/3 of that energy. So that you have the rest of the energy, which is this one, supposed to, for you to do whatever you want. Walk, dance, talk to the people, and do whatever you like to do. Well, not really, because part of it is just to be self-sufficient, just to walk, to be able to dress in the morning, go to the bathroom, and get dressed, get undressed, maintain the autonomy. And then part of this is due to the fact that instead of being in a perfect homeostatic equilibrium, sometimes you have disease. And when you have disease, they cost you energy. I mean, think about having an infection. You're going to spend a lot of energy by constructing those antibodies. And then you have a metabolic derangement. You have to use alternative pathways, alternative way to maintain the homeostasis that are going to be a lot more expensive. And then, not only that, so that you need an homeostatic effort due to the disease or aging. And then also, when you age, unfortunately, and I'll show you data, you actually need more energy to do the same thing. So if I walk from here to here, if I am 20, I'm using much less energy than when I'm 80. And that's really an important and hot area of investigation on why this is happening. So let's go through the data very quickly. So resting metabolic rate. Resting metabolic rate declines with aging. And so when I was studying and came to the BLSA, I looked at the data and I saw, well, there is a decline. The decline in part is due to lean body mass. And even when you adjust for lean body mass, there is some sort of decline with age. Is this good or bad? What do you think? Is this good or bad? Is decline in resting metabolic rate a good thing or a bad thing? Does somebody know when our resting metabolic rate is maximum? 2 days after birth. Because you are growing, and growing requires immense amount of energy. Protein synthesis is an exothermic reaction, requires immense amount of energy. So resting metabolic rate peak at 1 year, decline very, very slowly up to the age of 18, and then decline a little bit less, more slowly up to the age of 90 to 95. After 95, we don't have data. So this decline is shown here between the age of 30 and the age of 100. And I thought, well, the resting metabolic rate decline, so that low resting metabolic rate should be a risk factor for mortality, isn't it? Maybe yes, maybe not. And in fact, when I used this beautiful data that were collected before me, 35 years ago, the people in the BLS collected data on resting metabolic rate, I found that high resting metabolic rate is in fact a risk factor for mortality. And if you think about it, it makes a lot of sense, because if you have an homeostatic effort, if you're consuming a lot of energy to maintain the homeostatic equilibrium, it means that your homeostatic equilibrium is endangered. And so that is a very, very strong predictor. And then when you did longitudinal analysis in these people, we had multiple measures. And we look at where the split was. What was happening before that? So here in blue, you have those who are deceased, and in red, you have those who survived. And you can see that the split is 10 to 15 years prior to that. And those who died really don't show any accelerated decline in resting metabolic rate. So in a way, if we could really understand the effect and have normative data on resting metabolic rate could be an incredibly powerful parameter to understand that your health status is endangered and could be used as a very, very strong and effective way of measuring global health status in geriatrics. I want to show you this data because what I've been telling you is that when you look at longitudinally in the population, this is a trajectory of the BLSA participants. They were classified as healthy, ideal. We have a very strict set of criteria for healthy and not healthy. You can see that in the healthy, the resting metabolic rate adjusted for body composition declined much more than in the unhealthy individual. Now, let me walk you through this data, because these are quite interesting to me. So this you have seen many, many times. This is walking speed, cross-sectional data, BLSA, repeated in tons of studies showing that walking speed decline with aging, okay? And accelerate in old age. You know, here at age 80 in the BLSA, very healthy population, usually in the other population about the age of 75. That's what we see. Now, in this test, in the BLS, when I came in, I said, I want to study energy. So I measure oxygen consumption during walking. And what I saw was this data. So in spite of the fact that these people were walking much slower, the oxygen consumption did not show any effect on aging. And that suggests that if you express oxygen consumption instead of per minute or per second, per meter, which is the amount of work that people have done in that task, you see that the oxygen consumption actually goes up, showing that aging is associated with reduced energetic efficiency. So you need more energy to do the same thing. Why is that? I don't think anybody knows. But this is important, especially in older people and in frail people. Because if you are young and you have a tons of fitness, a tons of energetic capacity, if your walking costs a little bit less or a little bit more, it doesn't really matter. But when your fitness goes down and peak walking energy goes down, then your efficiency really can make a big difference between being able and not being able to walk. This is something that geriatricians intuitively know from a long time. I mean, the example I always give to my students is the fact that if you have a problem in equilibrium, a balance problem, for example, you have little strokes in the basal ganglia of your brain. What do? What is the clinical sign for that? The clinical sign is enlarging the base of support. If you don't want to walk and fall, you instead of walking like this, you're going to walk like this. And that is a clinical sign that every geriatrician should know. Because I'm enlarging the base of support. Instead, you know, if you play tennis, if you want to give a strong, strong shot, you have to you have to fold your knees, because that gives you so much low body centrum, large base of support. All the energy of your shot is going to go into the ball instead of going in other places, okay? Well, let me finish, and then I'll tell you if you study biomechanics, in biomechanical, people will always tell you that there are many, many strategies by which our walking is perfect and we consume as little energy as possible. One of the strategies, for example, and I can go through all of them, but one of the most important ones is that you consume most of the energy to put up and down your body centrum. So if you have to move your body center, move your body up and down, that costs you an enormous amount of energy. The least you have to move your body centrum, so the more stable you are with the body center, the least expensive is your walking. Now think about when I walk with a very narrow base of support. And you can see that I don't really move very much my head up and down. Not only, but now I'm walking really artificially, because the real walking is also a rotation of the pelvis. So I walk like this, I rotate my pelvis. So part of advancement I do is not extending my leg, but it's rotating my pelvis. And the rotation of the pelvis doesn't require any change in my body centrum. So I'm walking like this, and by doing this, my body center is almost always there. And so that's very energetically efficient. So let's think about what I do when I have enlarged my base of support. I enlarge my base of support, and I can't rotate my pelvis, because otherwise I will have to do something like that. And then also, because I cannot advance, I have to do this. Look at my head. My head is going down. So I have to go up and down, up and down. My walking become a lot more expensive. And so because it become more expensive, the efficiency, the energetic efficiency of my walking is gone. We don't consider that. Physical therapists do. If you do stroke rehabilitation, you get those incredible improvements in walking ability in the first 5 days. It's not because the neuromuscular system have improved, but because the physical therapists teach to the patient a much better energetic way of walking. And by doing that, they are improving their ability to walk because they have more energy to do it and more energy to compensate. So how do we study that? The way we study that, please.
I want to ask a question about running as opposed to walking, so it's going to be a little bit different. But in running circles, runners are always asking themselves, how come I can't run as fast when I was younger? I'm working as hard as when I was younger. Just things don't move as fast. And the two big ideas put forward are, one is your stride length shortens, so you're turning your legs over, but you don't go as far per stride. And the other is that your elastic recoil on your joints goes down. So a young runner hits the ground, and energy gets transferred into the joints to push for the next stride. But in old folks,
in a healthy individual, the limiting factor to running performance, I'm a runner, so I study that. I really like to read about it. It's cardiac function. You don't get enough blood into the muscles. So that is why at some point you cannot run stronger and you cannot run faster, because you can provide to your muscle enough energy. And so you start having an anaerobic metabolism, and when you are anaerobic metabolism, you create lactic acidosis, and then your performance goes down tremendously.
So the cardiologists say that your cardiac output doesn't decline as you age, and that's not really a factor. But
the cardiac is only one point, and that your cardiac output does not decline, but adaptation of the peripheral artery does. If you look at perfusion in the muscle, and you look at the microcirculation and the delivery of NO, that is dysregulated with aging. There is no doubt about that. So that's the key.
But your
I'm getting there. So why don't I show you something, and then you ask the question. Oh, okay.
Okay, just one to the previous slide.
Yes.
The oxygen consumption, it seems
like the panel suggests that with aging, you consume more oxygen with muscle.
Well, you consume more oxygen. I didn't say with muscle. I say you consume more oxygen.
So
That's very important difference.
There's a possibility to measure the organ-specific consumption, say,
You are absolutely right. I think that there is a chain from oxygen consumption to work delivered. There are many, many steps, and we need to look at each one of them, not just the utilization of oxygen in the mitochondria. That's what I'm getting to. And so that is the next slide. I don't know, I had something, I don't find it anymore, it's here. So so far what I've told you is that this one and this piece, how much oxygen, how much work performed. But in fact, there's so much going on in the middle. And in the middle there is, simplifying, because I could get much more complex than that, there is the cost of acquiring the oxygen from the environment, which is the intake, how much air you need to exchange in order to do the oxygen that you need for the circulation. And then you have the cardiovascular function and tissue perfusion. I think we know a lot about arterial stiffness and microcirculation, the macro arteries. We know almost nothing about adaptation to exercise with aging in the microcirculation, and is an active, very important area of investigation. We know, for example, that in PAD, that is the part that is not working anymore. And then you have that once you have been able to carry the oxygen there, you don't know whether the mitochondria are able to use that oxygen or not, and whether there is enough mitochondria there to do that. And then you don't know whether there is mitochondrial coupling and uncoupling that is affected by aging. There are some very little preliminary studies, but really not very much to see what's going on, and no longitudinal study to look at outcomes. And then, even when you have created the ATP, we don't know whether the ATP that you have is going to be used to do the same mechanical issue, because as I showed you before, there's so many biomechanical variables that are important there. And so we study, for example, we study each one of them. I mean, the new BLSA is really addressing each one of these components because we want to find out what is going on. And so that's why we select the measure. You know, it's not the typical epidemiological study. What we do, we have a paradigm, and we select the measure to fit that paradigm. And, you know, what I wanted to say is that is the uniqueness of the intramural research program. This could never be done with a grant, because it can never be done in 2 years. It needs to be done in 20 years. And probably I will not do it. Somebody after me will do it. But it's the only way we can really gather this information in the future. And so one of the things we do, when I came here, I implemented a complete state-of-the-art gate system. And I invite everybody to visit in Baltimore, because we have exactly the same system that Pixar has to do their movies. And what we do, we put these little markers in people, and then these biomarkers are seen by these infrared cameras. And there are always at least 3 cameras that see these points, so that using the coordinate, we can reconstruct the position of those markers in the 3-dimensional space. So we can reconstruct trajectory and velocity and angles of each part of the body. And so we can say what's happening to the people that have less walking efficiency. And I can show you one data. It's a paper that is now being published in Gait and Posture, showing that in walking, if you measure the maximum range of motion during gait, you see that there is an inverse correlation with oxygen consumption. So there is a change in gait that is probably due to the rigidity of the ankle that make people having to have much more movement in the knee that increase the amount of oxygen consumption. And that is why, you know, understanding what shoes we need to wear is going to be incredibly effective in understanding energetic efficiency during walking. And only, you know, if you put that into the general concept of walking efficiency, you can really do this type of studies. I don't know. How much time do I have? I want to show you, well, okay. So I'm totally jumping on homeostatic dysregulation. Will be a very, very long discussion, so maybe that's the next visit. But I want to give you some of the things we're doing on neurodegeneration in the BLSA, because I think we have truly some amazing data. So again, I am a lucky guy, and somebody collected data for me. So I could come here and show you this data. So Alan Zonderman and Susan Resnick and many other people started collecting data on cognition and on imaging in many, many individuals. And we have data on MRI. We look at volume, and we look at ischemic lesion. We are looking at MRA. We're looking at spectroscopy now, because in 2005, we implemented the 3T MRI directly in the BLSA suite. So we can actually measure them longitudinally over time. And then in a subgroup of the BLSA, we do study with PET, and we are looking both of the circulation part of the brain, but we're also looking at an oxygen consumption, but we're also looking at beta amyloid directly in vivo by using the Pittsburgh compound. And I think that we have some amazing data. This combined with the fact that many of our people, and more, because we're really pushing that as a part of the BLSA, go into autopsy. When they die, we do a complete autopsy, so we have tissue for each one of them. So one thing that we discovered, which I think is incredibly exciting, is that there were people that are asymptomatic Alzheimer's disease. So there are people that when they die, we do an autopsy, and we find massive, I say massive amount of beta amyloid plaques that are deposited in the brain. But when they die, their neurocognitive function was perfect, absolutely perfect. They did not show any sign of Alzheimer's disease, suggesting that, yes, amyloid is very important, but especially in humans, there should be some other resiliency mechanism that, when in place, avoid the beta amyloid to have the neurotoxicity that lead to Alzheimer's disease. And so we're currently studying this normal asymptomatic ID and people with the real Alzheimer's disease and MCI, that when they died were at both neuropathology, and when they died, they have a diagnosis of dementia. And we're doing many, many different things. One of the things we did, for example, in this study that we have replicated more recently, but was published in 2007, we look at, for example, at how big were the nucleus of the neurons in the hippocampus. And you can see that the nucleus volume in the asymptomatic Alzheimer's disease were much, much larger. And this really suggests some sort of compensation that was going on, and a lot more work, both in the genetic expression and methylation and acetylation area, have been done in the tissues collected in this population. One of the things I told you we're doing longitudinal trajectory of imaging with beta amyloid using the Pittsburgh compound. And these are probably